questions about this? This is a kind of a larger topic, this horizontal gene transfer. Just this idea that bacteria can do this.
Are
They can share genes.
plasmid and ectoplasmid transferred differently?
No.
Okay, so it's the same horizontal gene.
Yep. And they transfer plasmids through conjugation.
It's just one has the resistance.
Yes, this is a specific
Okay.
plasmid that has these particular ones. Other questions on this? And we know why mutations aren't usually that good, right?
Oh, for the missense and.
Yeah, missense is the only one that might potentially work, but all the rest of them, I mean, silent isn't even doing anything, and then, nonsense, no good for sure. Frame shift usually messes things up completely. So have you had a chance to try the worksheets? Yeah, okay. And once you do it a few times, it's very easy. It's just, you don't want it, like I said, you don't want the first time to do the exam.
Would you say that missense is one of the mutations that might work?
Okay, so missense mutation is what is seen for the new transpeptidase that MRSA has, the new topoisomerase that MRSA has, the new RNA polymerase that MRSA has, and we gave a specific missense for the Mycobacterium tuberculosis. So what's changed there is that the active site of the enzyme now has a slightly different amino acid, such that it makes that organism now resistant to those antibiotics, rifampin, penicillin, or even citral. Yeah. So that's the only one that maybe you can get away with, but it can be really bad too, like in the case of sickle cell anemia. And I think that worksheet kind of goes through that. So if the amino acid change is very different in size or polarity, the protein won't fold properly. And misfolded proteins don't have any function. So that's the issue there.
So the missense is responsible for the MRSA and MICO? Is that what you said?
So on there, we have, so it's on this one, I'm pretty sure. So we have the topoisomerase mutation, so this is slide 12.
Okay.
Okay, and then slide 13 describes what's going on with mycobacteria and their RNA polymerase. Both of these are missense mutations. Okay, so your question was on?
That was it.
Okay, all right. So, I don't have an image of the 5, right here. However, all the rest of them having an entire enzyme suddenly that knows to cut this antibiotic or add groups to it or pump it out, which really this is multiple genes here because it's more than one protein. Yes.
So, I'm sorry, I was just confused on what would cause the gene resistance. Would it be multiple mutations or would it be?
No, horizontal gene transfer.
Oh, okay, that's what I know.
Yeah, and of the horizontal gene transfer mechanisms, which is the most common? Conjugation. So transformation, it occurs, but only bacteria even know how to do it. So that's like really unlikely. It's not that it could happen that way, but it's very unlikely. The probability is low. But with conjugation, and in fact, that knowing that they can do gram negative to negative, gram positive to positive, and negative to positive, it's like, wow, this is really being spread, right? Transduction may or may not be, it's probably the second most common. But it's conjugation for sure, number one. Yes.
During vertical gene transfer, would mutations also occur there, or is it only more commonly during horizontal gene?
That's what I'm saying, yeah, there's definitely mutation here. It's 1 in every 10 to the 8th.
Okay, so it's
If they're in the
much-
presence of mutagens, it's gonna increase that
Like
rate
10
by
times
at least
or
10
more.
times or more. It could be like 100,000 times more. So, you know, when you're going, like in the hospital or in a lab and they have those signs that it sort of looks like a series of things, or at least not just 4 or 5, or in there drawn here. On this side, the lysogenic cycle, what we're seeing is that the bacteriophage lands, squirts in its DNA, and it circularizes, but now it recombines with the host chromosome and now gets copied right along with the host chromosome. These pictures make it seem like it's kind of a big piece that's being added, but it's not. They're really little. Remember how big I said that a plasmid is, just like 1 to 5%, so it's very, very small. So it's not really like noticed as being something that's extra or extra work. And so it's copied right alongside, and now it will go on its merry way until we get to the stress, right? Did we do that? Did we add the word stress over that? Okay. So stress, and at the stressful point, which for a bacterium, we already know what this is. If they're not in the correct temperatures and osmotic pressures and all of these things like that, then if they're on a nitrogen, carbon, all of these things, you're not a great host, is what the virus thinks. And so it excises, pulls out its DNA, and then just undergoes the lytic cycle. But this time, rather than one infected cell releasing many, it could be many, many cells, depending on how many times it's been copied right alongside. Yes?
For the 4B stage, would that cause whatever the host is to have like some sort of like altered state or disease type of state?
Well, no. I mean, that's the thing. It's at that point, when it's sort of silent within the cell, it's not expressing all of its contents. That's not happening.
So it's not really affecting it other than just being there, part of the DNA, and keep multiplying. Kind of like cancer, in a weird way, except causing
no.
symptoms.
No. I can't go there. But yes.
After it lyses, is the cell able to recover, or is it
No,
dead?
no. It's going to die because what it forces the cell to produce lysozyme. It's an enzyme that we have in all of our bodily fluids, and it cuts the peptidoglycan, and it makes a big hole.
Okay.
And I mean, it shows here as though it's just in one spot. It's all over the place. So
Where does the virus go from there?
from here to the next unwitting
To the next
cells.
bacteria or host?
Yeah, finding a host. Going to us, transduction. Transduction is the lytic phase, but then in their way to get out of this cell, they mispackage and take some host cell gene with them. That's what's being introduced to the cell next door. Okay, so this virus here is actually not a virus for real at all, because it's lacking all the genes to say make more capsid, make more viral DNA. It's lacking all of that, but what it did deliver is genes from the cell next door. Okay. Yes, Jake.
You might have covered this, but can two different bacteria share plasmids between each other?
Yes, that's conjugation.
Oh, okay.
That's this process here, right there.
Okay. I mean, two different types of bacteria, not the same.
Yes, so we know that can be pretty much any gram-negative to any gram-negative, any
Oh,
gram-positive
okay.
to any gram-positive, and any gram-negative to any gram-positive, but not this way. We haven't seen the gram-positive, gram-negative, not yet.
I have a question about that. So it's always like they form that connection and then the one forming the connection is giving.
Yes,
the
a
DNA.
copy, giving a
Copy.
copy.
So it never like forms it and takes something, it's always giving.
Yes, always giving, always a
Okay.
donor, and always a copy. That's important to understand because now they both have it. I do think of it like the vampire's kiss, just you're not one just because you passed it on. Do we have questions on some of the other, material?
Yeah, we had a question on microbial growth
Okay.
and kind of the 5 tubes and which aerotolerant anaerobes. It says the presence of one enzyme has a use. We got that. Sorry, microaerophiles, it was that one.
Okay, so I asked you to fix this
Okay.
for yourself. They express both.
Oh, okay.
They express both, but in low amounts. Not enough for a full atmospheric dose of oxygen. And I gave you the example that if, for example, they, like think of a situation, you might know somebody who it can have dairy, but not that much. And that's that. We just like they do express the enzyme, you know, beta galactosidase. Okay, but some people call it lactase. But they don't express the water.
Okay.
So they can't have a lot of dairy because then they just can't process it, and that's what happens here. So they have both enzymes, but just not enough for a full atmospheric dose.
And then we were trying to figure out, so facultative anaerobes also have both enzymes, but
Yes.
they grow throughout the tube where obligate aerobes want to be up top. Is that a difference with the enzymes?
Well, so there's another process going on down here, and there's planting,
Okay.
and aerobes can't. Yeast are facultative anaerobes. Yeah, if you try to make like beer or wine, you have to get rid of the oxygen first or they won't do it. Because they'll make a lot more ATP using oxygen than fermenting.
Along that same lines, for the superoxide dismutase and the catalase, you have on the next slide the formula for it. Are we gonna need to know that formula per se?
No.
Or to recognize it?
No. I mean, we're gonna do this test in lab and then we'll talk about it in more detail. Just kind of like this.
Or no, 24, sorry.
Yeah, I mean, we're gonna do this in lab.
Okay. And for the test tube, kind of representing that chart, are you gonna be using the ones you want, the reference to like slide 25, where it's physical pictures of test tubes?
I can't because it won't, I've already tried this, it won't Xerox properly.
Okay.
It won't copy properly.
So we're good about it, so
I tried, it just won't, it doesn't work. It doesn't work, then you can't see who's who, it just doesn't work. So I have to use this is,
Okay.
I mean, obviously this is superior, but I can't copy it. knowing about it, yes, but also understanding that we're looking at all of this based on how antibiotics affect these processes of replication, transcription, and translation. Antibiotics, of course, lots and lots there, there's an entire table you have to be able to produce. Okay, so you wanna be able to get that together, and, it's 15 points in the bucket if you can do that. Okay, and then, antibiotic resistance, and now we know how they get that. And we understand about the different mutations that they could potentially make. Most of the time it's not gonna work. This is why they do this. This is the reason why, because the only other way they could get genetic variation is to mutate, which doesn't really work out normal. And that's it. So not too much there. I know it seems like we spent a lot more time leading up to exam 1, but we really do need to go like, okay, what are microbes? What do they do? What do they look like? So that we have to spend a lot of time like building up. And now once we're in here, I mean, the next exam after this is 2 topics, metabolism and virus, that's it. Because they're both dense, and that's enough. Okay, so,
So I'm sure it's somewhere in my notes, but can you refresh your memory on which day we were talking about for the 15 points?
so,
I'm trying to find it.
It's the antibiotics where it's like therapeutic, control, and spectrum.
Yeah, okay.
And then we just needed to pick one from each class,
yeah,
not including penicillin because we already have the.
right.
One antibiotic.
Do we need to know how to actually draw the mRNA, tRNA, and RNA? Well, there was a thing on one of the study guides that said
No,
practice
no,
drawing.
no.
Okay.
Oh, we didn't get this.
Well, I think we'll just. Yeah.
That's what we
Yeah,
did
drawing
today.
labor,
Yes, but not like draw the structure, I mean, like, just across from an A, you go either a T or a U, depending on if you're making, DNA or.
we were actually.
That's why I was like, do I need to do this?
Yeah, so you drew it, how did you draw your muscle?
Oh, like drawing out like the phosphate and then.
You did?
Yeah.
Well, you don't need to do that.
Okay.
We don't do structures in here, I mean, I draw structures because I can't understand anything unless I see it.
Well,
But-
it's sugar phosphate.
Yeah.
Do we get to know roughly how many like multiple choice or
No, I haven't made up the exam. I have no idea. I mean, the exam 2 review is an excellent, gauge on what it might look like.
Okay.
No, I've not made up the exam. I don't like to do that because then, and I know many people do it that way, many instructors do it that way, but I don't do it that way because then I will consciously or unconsciously be teaching to that test, and I don't like that. I say this to you guys all the time, that I am the boss of this, not the other way around. I recognize you have to have something. I can't make up a review sheet on the fly every time, so I gotta have something, but, I can't let that determine what's going on. Let the lab book tell me what's happening, it's like, it's what's new and current for us, so.
So just to clarify, for the, how do you say, micro aerophiles,
Yes.
they produce both the SOD and the catalyzed enzyme, but just in very low concentrations?
Yes.
Okay.
Yeah, that's not written on here.
For the RNA stuff and DNA stuff, are you gonna format it similar to the worksheet? Or will we have to figure out 5' to 3' and then going from 3' to 5'?
Well, I mean, what do you mean?
So on the worksheet, you just have written out, it's not 5' to 3' or 3' to 5', it's just, there's no primes on it.
Oh, I will give you
Well,
that.
You will get,
Oh,
okay.
yes, yeah.
Okay.
Maybe I didn't notice it, but is there an answer sheet up somewhere for
Mm-mm.
the
No, I don't
worksheet?
have that.
Okay.
I'm happy to go over it with you, if you want, but
Okay.
no, I don't.
Can we do it now?
Sure. Anybody has a question?
I was just gonna say, for the obligate aerobes and like facultative anaerobes, they both produce both types of enzymes. Is there a difference in the amounts they produce of each?
And these two,
Yeah, like they both produce both types of enzymes.
the truth is, I don't know what to tell you about that. I mean, they can both do it and they are both happiest with maximum molecular oxygen concentration, so I assume their expression of that must be really similar.
The difference is the facultative ferments. That's why it's able to grow down where there's less oxygen.
Is it ferments?
Yes.
Okay, so you wanna go over now?